Hi, my name is Helen Willsey. I'm an assistant professor at UCSF and an investigator with the Chan Zuckerberg Biohub in San Francisco. And today I'm going to be telling you about our work tackling KCNT1-associated comorbidities using a frog model. So buckle up. We use frogs because at any given moment in my lab, we're studying about 100 different neurodevelopmental disorder associated genes. And so we wanted a high throughput scalable platform to go after many of these genes in parallel. So we chose diploid frogs, which is Xenopus tropicalis, as our model organism of choice because it is high throughput. Unlike zebrafish that have a duplicated genome, frogs do not. They have a simple diploid genome. And a single mating pair, which are these guys you can see right here, will lay over 4,000 embryos in a day. This dish here is actually a 15-centimeter dish, and each one of these dots is a fertilized embryo that we can manipulate. They have rapid development in just 7 days. You have a free swimming, behaving, eating, pooping, moving tadpole. They're very cost effective. We can do drug screening just by putting drugs in the water with the tadpoles in 96-well plate format, so we can scale up for drug repurposing. But if you remember one thing about frogs, let it be this. You can inject at the 2-cell stage and just manipulate half of the embryo by injecting into one cell of those 2-cell stage embryos, and that ends up being inherited in just half of the tadpole. And so we can create these animals where you actually have the internal control within the animal itself and compare the left to the right half of the animal to understand what that gene's function was. It's high throughput, it's quick, and it's cheap, $25 per gene. And here we're showing an injection of a CRISPR with a dye, but of course you could also overexpress something, put in a different allele, do all sorts of different manipulations, and whatever you can dream of to inject. The embryos are very robust, and so you can overexpress things pretty easily. So a typical day in my lab we were creating thousands of half manipulated animals and then seeing what happens when we manipulate gene function. One of the things that we've started to get really interested in, because it seems like a hub of convergent biology among neurodevelopmental disorders, is a subcellular organelle called cilia. And so cilia are present on almost all cells of the body, including neurons. They come in 2 flavors. So one type of cilia are motile cilia. So it's these hair-like projections that come out of the surface of the cell. And in this case, these cilia actually physically beat and create fluid flow. So these are the kind of cilia that are in the respiratory tract that are moving mucus and providing a barrier to infection. These are also the kinds of cilia that are in the ventricles of the brain, moving CSF fluid. These are also the cilia that power sperm to move, etc. All other cell types, with only a few exceptions, have what's called a primary cilium, which is this small pink antenna-like structure that comes out of the cells. And so almost all neurons have them. They're very important for regulating neuronal excitability. And they're very important for neuron migration, as well as many key developmental signaling pathways like hedgehog signaling, very important for the development of the heart and the kidneys, as well as many other things. And there are common disorders, multi-organ disorders that are caused by cilia defects and comprise a whole class of what are called ciliopathies. And so you may recognize many of these disorders as present in the KCNT1 population. I'll show you some of that in a second. But of course, cilia defects on their own can cause epilepsy. They can cause heart defects, kidney defects, and very severe respiratory problems with overproduction of mucus, etc. In terms of chronic respiratory disease is where you usually see a lot of these symptoms. Frogs just happen to be an excellent model to study cilia. 48 hours after fertilization, the frogs have a ciliated epidermis where they have all these individual ciliated cells, each one putting up 200 cilia that are physically beating and moving mucus along the surface of the frog's skin. And so this is a fantastic model system for the human respiratory tract, because the human respiratory tract is very similar to this, having spaced out motile, multiciliated cells moving mucus. And so the real question we got interested in here is, does KCNT have a role at the cilium? Could that explain some of these comorbidities that we're seeing? We were especially intrigued by this because there's evidence that KCNH1, albeit a very different channel, but the idea that potassium flux at the cilium may be important for developmental biology, we got excited about. So the graduate student that's been doing this work is Angeline Chemmel. So everything I'm about to show you was done by Angeline. So to see if this was even possible, we first wanted to look at the citizen health data from 46 patients with KCNT1 variant. So thanks to Citizen Health and for all the families that participated in this medical record review. And so what you'll see is within these 46 individuals, almost all of them have seizures, very severe respiratory issues, but also skeletal defects, cardiac issues, kidney issues, dysmorphic features, again, all plausibly have a ciliary contribution. And so that at least was consistent with the possibility that cilia could be involved. What was interesting with the cardiac issues is, of course, if this was a neuron-only mechanism, we would expect maybe arrhythmias, and those, of course, are there. 15 out of the 46 patients reported arrhythmias, but there are also things like congenital structural heart defects that are hard to explain by a neuron-only mechanism. Congenital structural heart defects are often caused by motile cilia problems in the early embryo. And also 22 of these 46 patients had heart murmurs, again, a structural problem in heart development that's hard to explain by a neuron-only mechanism. Dysmorphic features, which are really about the facial structures, are also very, these ones, the 10 patients out of 46 facial structures, those often arise from neural crest issues during development, which are very highly involved, cilia are very highly involved in the migration of those cells. And then of the reported respiratory issues, of course, many of these patients are on assisted breathing. But this excess fluid was very intriguing to us, and cilia, of course, are very important both in the generation and the movement of fluid in the respiratory tract. But there were also 15 patients with structural defects in the respiratory tract, which again are hard to explain by a neuron-only mechanism. So we first looked at the early embryo to see where KCNT1 is expressed over development. Again, if it was only in the neurons, you would only see neuronal cells, but we actually see very broad expression of KCNT1, both in the neural tube that does become the brain, but also in those neural crest cells that end up forming the facial features. We also see it in the ciliated epidermis, that punctate pattern along the skin of the frog, in the highly ciliated cells. We also see in the otic vesicle, which becomes the ear, which is highly ciliated, and the eye, which is highly ciliated. And a little bit later in development, you also see it being expressed in the developing kidney, here labeled as the pronephros, and then also in the developing heart in the early embryo. So it looks like there's very broad expression of KCNT1 in developing embryos, and that it's largely in ciliated tissues. And so the first thing Angeline did was create a loss of function. So obviously the mechanism of pathogenesis is gain of function, but we wanted to first establish what is the baseline function of KCNT1 in cilia. And so to do that, she created a translation blocking morpholino that targets KCNT1 expression in the early embryo and injected it into the frogs. In the control situation, the green is tracing which cells got the morpholino. And you can see in the control situation, like I showed you earlier, is that if you look at these cilia, which are these pink dots along the whole embryo, that's the head, that's the tail, you see these very nicely spaced, regular multiciliated cells with nice cilia. But when she knocks down KCNT1, you start to get these entirely bald patches where you lose the formation of cilia. And this is incredibly reproducible. Over 47 embryos she has scored now for each condition. And these are a severe loss of cilia in these animals. We also wanted to then see if we could phenocopy this with another way of manipulating KCNT1. So she took advantage of a KCNT1 chemical antagonist, this drug here, and again applied it to embryos over development. And same thing, these are a little bit later stage tadpoles, but when you look at a DMSO control embryo over the entire skin of the frog, you see these beautiful multiciliated cells with nice cilia. But then if they're instead raised in this antagonist, you lose this cilia completely, which is not a typical phenotype you see with drug treatments. And again, she scored this over 120 embryos. So it's a very reproducible phenotype where we see this total loss of ciliation. That's where we're working now on the downstream mechanisms and why we think KCNT1 is important for normal cilia formation. And we think that this will be helpful for us to understand the mechanisms of its role at cilia, to then understand what's downstream of it and thinking about gene therapies and repurposing drugs to go downstream of KCNT1 to reduce gene function in a gain of function model. So with that, of course, I want to acknowledge the lab, especially Angeline, who is pioneering this project, and Citizen Health, who provided the medical record data, and families that are participating in research. Thank you so much. And our funding agencies and our Xenopus resources, and especially KCNT1 Foundation and Allie. We've had so many fantastic conversations and thinking about how to be responsive to patient priorities and these comorbidities. And when we're thinking about cilia, maybe I haven't been explicit about it, but we're really thinking about the respiratory issues that these kids are facing and also the GI issues. And so I didn't talk about it today, but cilia are really important for the migration of neurons into the gut and the enteric neuron development. And so we're also have another project thinking about that with KCNT1. So thanks for your attention. Happy to take any questions.